Well, welcome everybody to the fourth session of this series, which we're focusing on aging. Today, we welcome Nick Lane, and we're going to ask Rhys, who's been carrying out research for us, Rhys Mould, at University of Westminster in the Optimum Health Labs since 2018. So Rhys will be introducing the talk and introducing Nick today.
Good morning from California. My name is Rhys Mould. I'm, as Jeffrey said, a postdoc for the Guy Foundation, usually at the University of Westminster. And it's my great privilege today to introduce today's speaker, the fourth speaker in our spring series on aging, Professor Nick Lane from UCL. In his field, Nick has published over 100 peer-reviewed papers, and you also might know him as the author of 5 acclaimed books, including Transformer, Vital Question, Life Ascending, Power, Sex and Suicide, and Oxygen, the Molecule That Made the World. Together, the books have sold over 150,000 copies worldwide. On top of his writing, Nick has also earned numerous prizes and accolades for his work, including the 2015 Biochemical Society Award and the 2016 Royal Society Michael Faraday Prize for excellence in communicating science. Nick's research interests dwell within evolutionary biochemistry and bioenergetics, and in particular, 3 major transitions in evolution: the origin of life, the origin of the eukaryotic cell, and the evolution of conserved traits such as sex, speciation, and senescence. Between each of those transitions, Nick believes bioenergetics govern each transition. And as we know, governing bioenergetics is the mitochondria, which brings us on to the topic of this presentation. Mitochondria and aging. We know that mitochondria are responsible for energy production, cell signaling, cell death, and they also play a huge role in aging. But what we don't really know, or what's more unclear, is how do they change during aging? Now, the mitochondria have a long history with aging, notably as part of the free radical theory of aging. We know that mitochondria produce reactive oxygen species, or ROS, as a byproduct of respiration. ROS are used as signaling pathways, and the cell can't live without them, but the accumulation can lead to toxic side effects. The free radical theory of aging says that the accumulation of these ROS, the prolonged accumulation of these ROS, is what leads to aging. But the picture is a little more complex, and the theory has evolved and grown over time. And what we're trying to figure out is, what is it that causes this prolonged accumulation? In today's talk, Nick is going to home in on complex 1, which is the first in the respiratory chain within the mitochondria, NADH reductase. And what we're going to be looking at is how exactly is it that this complex 1 changes with age? How does its function, morphology, size, etc. change as the organism ages? And then we're going to zoom out and also look at how the arrangement of mitochondria changes during aging. On the left, you see the mitochondria arranged in their long networks. And then if you zoom in to the right-hand side picture, you can see the cristae of the mitochondria, the invaginations in the membrane, which is where the respiratory complexes sit. And what we're going to be looking at today is how the structure of these cristae change and how that might be related to aging. So before I give it all away, I'm going to hand over to Nick for what I think promises to be a really fascinating talk.
Well, thank you very much for the invitation. I've given a very deliberate kind of conversational tone to my title, because this is, as some of you will appreciate, not what I spend most of my time working on, but it's something I've been very interested in over decades. And I do actually have one guy in my lab, Enrique Rodriguez, who's been working on mitochondria in Drosophila in aging, and so I'm going to show some of the results that he's got. But I'm unable to interpret most of these results. I want to kind of show you the kinds of things that we've been wrestling with and how I'm trying to think about it, and perhaps a few of you will be able to come back afterwards and and correct me and give me better ways of thinking about it. Anyway, so It's also, I have this picture of Warburg. This is, actually it was last year, was 100 years since he published his first paper on what's become known as the Warburg effect. And it links specifically to cancer, but really to the idea that cancer is far more common in as we get older, and it's an age-related disease. And he saw it as an age-related deficit in respiration. He saw it as a reversion to some kind of primitive cell type, like to a yeast-like phenotype and fermentation. And it's been a very controversial idea over literally 100 years. But there is something in it, and there's been more or less an exponential increase in the papers citing the Warburg effect over the last 20 years or so. And really, I think most people, in thinking purely in terms of ATP and energy flow in cells, have misinterpreted it. This is not a particularly original idea, but I think it's far more closely linked to Krebs cycle flux and epigenetic state and the use of Krebs cycle intermediates as precursors for biosynthesis. So that leads to a permissive cellular environment that increases cancer risk steeply with age. Let me just see if I can put a pointer on here. So I'm going to give a broad overview of my take on this. I'm going to come back to it at the end, and especially respiratory complex 1, which I'm going to try and present probably rather controversially as an electromagnetic engine. So I'm going to return to that. But first, before I get going, I want to give a quick perspective. Most of my work, as I say, is on the origin of life. And it's been really rather a surprise to me to discover that many of the themes that I've worked on with the origin of life and with cancer and aging are dealing with essentially the same set of actors. So our approach to the origin of life has really been to use life as a guide to its own origin and to try and get the granularity right. And the granularity, we've been thinking in terms of 3 effects. One of them is energy, one is metabolism, and one is genetics. So that's a very, it's very granular indeed. And I'm trying to get the moving parts in place. So this is very much a cartoon of what's happening in respiration. We are taking glucose or any other fuel, breaking it down to acetyl-CoA, going into the Krebs cycle. And the Krebs cycle is effectively spinning out carbon dioxide and hydrogen. Now, I'm specially saying hydrogen or 2H rather than NADH because it's really the hydrogen atoms that are the important thing to keep in mind here. Those hydrogen atoms are split into the protons and the electrons, and there's in effect a current of electrons within the membrane flowing to oxygen. Doesn't have to be oxygen, it can be other electron acceptors, and there's plenty of anaerobic bacteria out there that have terminal electron acceptors that are not oxygen, but the overall topology and processes are maintained. They're exactly analogous in that. So we have a current of electrons to a terminal electron acceptor. That powers the extrusion of protons across the membrane, giving an electrochemical proton gradient across the membrane. That is both pH gradient and a membrane potential, because protons obviously are positively charged. So that is the protonmotive force, the influx of protons back through the ATP synthase, which I'm sorry I missed Wayne's talk, I think, last week, but you would have no doubt heard plenty about that. Driving ATP synthesis. It's important to say, though, not only ATP synthesis, it drives work more generally. So again, I'm trying to keep this granularity. This is what's happening on our own mitochondria. We could see this as the inner mitochondrial membrane, but this is also happening in bacteria. And I think this kind of process, not necessarily with oxygen as an electron acceptor, but this kind of process goes back to the origin of life. This is not the same, it's the opposite. What you're seeing here is the reverse Krebs cycle. Now, most people in biomedical research have really historically, this was discovered in 1966. It took a long time before anybody accepted it was real. And even today, I don't know that medical students are really taught anything about the reverse Krebs cycle, which is found in plenty of bacteria and is possibly really very widespread in archaea as well. It's not absolutely clear how widespread it is. It's not always a complete Krebs cycle. That's the first thing to appreciate. But what we're seeing here, this is why I'm stressing hydrogen rather than NADH, we're seeing a membrane potential driving the reaction between hydrogen and CO2 to form Krebs cycle intermediates. And this is a reverse Krebs cycle flowing in the opposite direction. And from these Krebs cycle intermediates, carboxylic acids, bacteria are forming amino acids, sugars, nucleotides from there, lipids, basically everything. The reverse Krebs cycle is the heart of biosynthesis. It's slightly longer than the oxidative Krebs cycle that we all know. It includes pyruvate and it includes phosphoenol pyruvate and so on. So if you're thinking in medical terms that that's part of glycolysis out in the cytosol or something, not in bacteria. This is all part of the reverse Krebs cycle and is all about CO2 fixation using hydrogen taken from the environment. And it requires energy, and that energy is produced by, again, the proton-motive force. I'm showing here something equivalent to methanogenesis. This is the simplest way of doing it, where that hydrogen is reacting with CO2 to form methane as a waste product, but also reacting with CO2 to form the precursors for biosynthesis. So this is the heart of metabolism as we know it, and we're keeping these two factors together. In this case, the proton-motive force is driving work, and that work is CO2 fixation. It's not necessarily ATP synthesis. And this is the third of these three components, information. There are patterns in the genetic code that have been known about since the 1960s, and they've proved quite enigmatic. And if we reinterpret them in relation to metabolism, to an autotrophic metabolism, they make a lot more sense. Now, these patterns, there is a relationship with metabolism itself, and there's also relationships, I'm not going to go into any detail on this today, but relating to their size and the hydrophobicity or some related physical properties of amino acids. Implying, at least at a top line level, if you had a random sequence of RNA, then you would have interactions with a non-random set of amino acids. We don't know if it's in a small binding pocket of an oligomer, you might call it a proto-RNA, a proto-transfer RNA, or if it's with something that looks more like messenger RNA. But the point is, these are non-random associations, and if you are able to polymerize them, you could have a completely random RNA giving rise to a non-random peptide, reliably so, statistically, but reliably. And that can have function in certain environments. In other words, it's a very easy way of bringing information into biology. That implies that proticity, which is to say proton-motive force, drives metabolism, which gives rise to genes. And from an origin of life perspective, that implies that everything's happening in one place. That seems perhaps a little improbable, but there are places which make sense. Alkaline hydrothermal vents is the one that I have spent 10 years working on. But not everybody agrees with that, and I'm not going to be talking about that in this lecture. This, though, is essentially our hypothesis for the origin of life in a nutshell. We have in these vents, there are inorganic pores. Those pores have got very often catalytic walls with iron sulfide minerals included in those walls, which have a structure very similar to the iron sulfur clusters in proteins like ferredoxin that are still responsible for CO2 fixation. We have natural proton gradients. The early oceans were acidic. These alkaline hydrothermal vents have got alkaline fluids, and so we have a natural proton-motive force driving CO2 fixation with hydrogen. These vents bubble with hydrogen gas and CO2 from the oceans, giving rise to the thermodynamically most favored products, which would be fatty acids, I'm putting the lipids here, and amino acids. The lipids, in principle, could form a bilayer. In practice, we've done a lot of experimental work, which I'll perhaps touch on some of this in my talk in the autumn. But the key point here is I'm just showing topologically that you can have protocells inside these pores, inside hydrothermal systems. We now have a partially closed system, but still partially open, driving not just lipids and amino acids, but potentially sugars and nucleotides, and eventually an RNA world where the genetic code first appears all inside protocells, inside hydrothermal systems. Now, I'm telling you this in relation to aging. You may wonder why. Because processes like the Krebs cycle and the protonmotive force are provided effectively by the environment. They are not, they're not simply a product of genes. They are not an invention of information in the world, in biology. They are something that geology provides for free. All of these conditions can lead to a protometabolism, which is topologically equivalent to metabolism, at which the heart is the Krebs cycle. And genes arise from that, and genes then modulate all of that. Just to, again, I don't want to go through any data on the origin of life today, but just to give you a sense of the lie of the landscape here, if we start with CO2 at the top corner, this is the chemistry of hydrogen and CO2. In brown here is hydrogen, hydrogen, hydrogen. There's hydrogen everywhere you look. In green, it's CO2. We're going through molecules like methyl groups and carbon monoxide, but very quickly we get to not necessarily acetyl-CoA, but an acetyl group into pyruvate, oxaloacetate, alpha-ketoglutarate. These are Krebs cycle intermediates into sugars, to glyceraldehyde. And then amino acids like aspartate, for example, glutamate, glutamine, they're very, very close to. It's only a handful of reactions from CO2 fixation before you're into these Krebs cycle intermediates. And we add in some ammonia, and we're getting amino acids like aspartate. In the red boxes here, these are the precursors for nucleotide synthesis. So what I'm trying to suggest here is that metabolism is so deeply conserved, it's older than genes. This is something that happens spontaneously in probably multiple environments, but certainly in this kind of environment where hydrogen is plentiful, CO2 is plentiful, a little bit of ammonia around, and so on. In blue, the blue arrows here are things that have been done in the lab under plausible prebiotic conditions. We've done this step since then to make aspartate. Pretty much all of them have been done in one way or another now. In fact, this step to make ribose phosphate as well. So pretty much all of this has been shown to be spontaneous chemistry that can just happen. This is the last thing I'm going to say on the origin of life before moving on to what it all means. What one can imagine is some kind of protocell inside a hydrothermal vent where we have now a bilayer fatty acid membrane through which protons are driving some kind of protometabolism similar to what I've just shown you, forming amino acids and nucleotides, nucleotide phosphates, or nucleoside triphosphates. Polymerization gets us through to peptides and short RNA oligomers. We have stereochemical interactions. We end up with something that looks a little bit like a proto-translation, and then various feedback loops on CO2 fixation, on polymerization, on protometabolism, and so on. So this is very much a structure for how we're thinking about metabolism and how things come together at the origin of life. And really, my main point in relation to all of this is that the Krebs cycle is at the heart of it all, and that it is modulating what genes are doing. And has been from the very beginning. So I don't blame you if you think that this is a bit hand-wavy in terms of linking the origin of life with aging, but I would like to try and get across to you why I think it does still matter. So we're going to fast forward by about 3.5 billion years to the Cambrian explosion. And this is a lovely piece of art from Miller, paleo art. On the left-hand side here, this is the Ediacaran fauna from around about 560 million years ago. And on the right-hand side, this is the Cambrian animals from around about 540 million years ago, or a little more recently. This is the Cambrian explosion, it's very often called. And before the Cambrian explosion, these are animals, but they're almost certainly sessile filter feeders fixed to the spot. They don't move around. They don't do anything very interesting. They look a lot like plants, but they seem to be fixed to even the deep oceans. And so the usual interpretation is they're more like sponges or corals. These are filter feeders. The one thing we can be sure about, though, with the Cambrian explosion is, we're now seeing eyes and claws and armor plating and all these kind of things. This is a landscape of predators and prey and complex ecosystems that are familiar to us today. And we can be absolutely certain then that they had mitochondria and were using those mitochondria for aerobic respiration, because nothing else is really capable of enabling ecosystems with 4, 5, 6 trophic levels, which allows predators and prey to exist at all. So if you've got a strictly anaerobic system, under fairly strictly anaerobic conditions, it's not just fermentation. There's all kinds of forms of anaerobic respiration, so using nitrate or nitrite, for example, as an electron acceptor, but they simply don't accumulate in the absence of oxygen. So ironically, as soon as you get oxygen building up to any levels at all, you also then get an excess of anaerobic electron acceptors, nitrate, sulfate. All of these things become far more available in the presence of oxygen than they ever were beforehand. So these early anaerobic systems were very limited and you simply, there's not enough energy in those systems. This is really related simply to the energy conservation between trophic levels. It's around about 10% maximum efficiency for things like fermentation, giving you about 2 trophic levels before you're practically out of energy. Whereas around about a 40% efficiency with aerobic respiration gives you 5 or 6 trophic levels before you're down to that same energy level. So extended food webs are possible with aerobic respiration, and we can be virtually certain then from the appearance of these animals and their behavior and so on that they were aerobic, that they had an oxidative Krebs cycle, and therefore we can be fairly certain that they would have been aging as well in a way that would be recognizable to us and would have suffered from diseases age-related diseases if they survived that long. And that leads to an interesting question, which is to say, the first few billion years of life on Earth, certainly the first 2 billion years, is dominated by what amounts to a reverse Krebs cycle as the engine of biosynthesis, giving rise to lipids, amino acids, sugars, nucleotides, and so on. And then from the time of the Cambrian explosion onwards, I don't know what you imagine. I imagine it kind of screeching to a halt and turning around and going in the opposite direction. Obviously, it didn't do that, but there's then the question: so what did it do instead? The chemistry, of course, is interestingly similar, but one of them is going to thermodynamically dominate in oxygenated atmospheres, and this is going to thermodynamically dominate in anaerobic atmospheres. So how do you go from one to the other one? Well, for most bacteria, actually, it's never a Krebs cycle, and in many of our own cells, it's never a Krebs cycle either. It's it's more of a roundabout with certain things coming in some places and leaving in other places and so on, and you can have flux in both directions through different bits of the Krebs cycle. But just to try and simplify this, what we often see in bacteria from acetyl-CoA is CO2 fixation to pyruvate, through to oxaloacetate. And there we can have an oxidative prong going through to alpha-ketoglutarate, as we see in the modern oxidative Krebs cycle, or alternatively and simultaneously, we can have a reductive prong going through to malate, fumarate, and succinate. And so it's not a complete cycle at all. We just have 2 prongs, one oxidative, one reductive. And this is all about NADH balance. This is NAD to NADH ratios. It's keeping redox balance in cells. And this little bit here between alpha-ketoglutarate and succinate, it can go either way. This is very sensitive to the actual redox conditions in the environment. And so ultimately redox balance determines which direction flux takes across there. And that's still central to how many animals work. So, for example, turtles that can often hold their breath for a couple of hours underwater. This goes back to Peter Hachachka's work with Ken Story in the 1970s. And what they had realized is that complex 2, under anaerobic conditions, fumarate will act as an electron acceptor. and can accept it. So the enzyme here to generate any energy from this, it's not succinate dehydrogenase, it's fumarate reductase, but effectively electrons are being transferred from NADH in complex 1 to fumarate in complex 2, generating succinate as a waste product. And there is enough energy in that to pump 4 protons, and that can drive, not much, but apparently some ATP synthesis. You require the fumarate reductase enzyme to do that. We also do this with succinate dehydrogenase. We can't generate any energy from it, but this happens during ischemia. And so ischemia meaning not just things like organ transplantation, but myocardial infarctions and cerebral infarctions and so on as well, where blood flow is cut off for a while, succinate will accumulate. And on reperfusion, that can then be oxidized through a normal oxidative Krebs cycle back to fumarate. So remember, this is the key to redox balance in bacteria, and it's still the key to redox balance in many modern animals. And it's a big problem in ischemia reperfusion injury because it will drive reverse electron flow. Effectively, there's more driving electrons into complex 2. The redox span between succinate and oxygen is very similar to the redox span between NADH and oxygen, but you're only pumping 6 protons instead of 10 protons, so you generate a higher proton motive force, and that can drive reverse electron flow into complex 1 and will form NADH at complex 1. Very often ROS as well, reactive oxygen species at complex 1. So this is something which is real. How common it is another matter, but it's not simply an accident here. This is about maintaining redox balance and determining. And succinate can leak out of the mitochondria and will have all kinds of effects on, for example, HIF1 alpha and gene expression in relation to changes in oxygen levels. So here's the respiratory chain. Here's the Krebs cycle. Is generating NADH. That's being oxidized primarily and almost completely in complex 1. Electrons, I've not done a great job with this figure, but electrons are traveling through to complex 3, to the Q cycle in complex 3, through cytochrome C to complex 4. There's 4 electrons being pumped here, 4 at complex 3 and 2 complex 4, and then the flux of electrons, as you know, coming through the ATP synthase is driving ATP synthesis. Now, when most people who work on mitochondria think about damage to the respiratory chain, primarily what we think of is lower rates of respiration. It's true, and there can be lower rates of respiration if there is damage to or mutations or whatever it may be in complex 1, for example. But perhaps almost equally importantly, that means that NADH is no longer oxidized in the way it normally would be, and so can build up. And because NADH is being produced by all of these Krebs cycle steps, as it builds up, it will tend to drive them in reverse. And so various parts of the Krebs cycle will go into reverse. And we can see that in many conditions. Citrate very often is exported to the cytosol. And I'm pointing out ROS here because if we have a highly reduced complex 1, but electron flow is not really getting much further down the respiratory chain, then the quinone binding site, and very often the NADH flavin binding site here are the main centers for ROS production from complex 1 coming in to the mitochondrial matrix. Cancer, one thing to say about this is we're beginning to set up something that's looking like the Warburg shifts. So we have damage to complex 1, we have apparently damage to respiration, but very often this can be compensated for by direct electron input into complex 3, for example. And we have a Krebs cycle that is beginning to turn to generate citrate, which can go out to the cytosol. Many cancers love glutamine, and glutamine is broken down. It's deaminated to alpha-ketoglutarate. And alpha-ketoglutarate, this is a kind of a slightly different view of the Krebs cycle, But flux can go in both ways from there, and in fact, it's linked. It seems to need to go in both ways, and this is, I should have put a reference on this, but this is work in cancer as well. What you're seeing here then is it can be decarboxylated to succinyl-CoA, and that generates NADH, and from succinyl-CoA to succinate, that's generating ultimately ATP. It's actually GTP, but you go through to ATP in the end. And succinate then is very often leaks out from the mitochondria. It can also be carboxylated with the carbon dioxide from here to form isocitrate, and that requires NADPH. So where does the NADPH come from? Well, it comes from the transhydrogenase in the membrane, and that is transferring, it's using the protonmotive force to power the transfer of hydrogen from NADH onto NADP to form NADPH. So cancer cells very often require a membrane potential. They don't really need it for ATP synthesis. What they really need it for is NADPH to generate citrate, and citrate is also exported to the cytosol. And so we have a kind of a flux from the mitochondria of citrate and succinate. This is far from Krebs cycle behavior or oxidative phosphorylation as we know it, but it still requires a proton motive force to be driving any of this. In the cytosol, I'm just touching very briefly, citrate can be then broken down again through Krebs cycle enzymes like citrate hydrogen lyase in the cytosol to form oxaloacetate and acetyl-CoA. So this is the opposite of what's happening in the mitochondria. Oxaloacetate is required for gluconeogenesis. It's required for nucleotide synthesis. So it's really the heart of making more nucleotides for cell growth. And acetyl-CoA is required for fatty acid synthesis, so new cell membranes and so on. But also in terms of epigenetics, sirtuins, for example, are switched off because of histone acetylation. And plenty of NADH around means sirtuins are switched off, they're not deacetylated. So this is beginning to switch the epigenetic state of cells. Succinate as well, I mentioned HIF1 alpha, that tends to drive glycolysis, to switch towards glycolysis away from oxidative phosphorylation as a source of ATP. And also the ingrowth of blood vessels, angiogenesis, inflammation, and so on. And so these two Krebs cycle intermediates are beginning to press an epigenetic switch towards something that you could say looks like a senescent state. This is really driving growth. This is the mTOR axis of cell growth. You're suppressing the sirtuins, which are responsible for stress resistance, and you're upregulating mTOR and so on, which is responsible for driving growth and proliferation, as does angiogenesis and inflammation and so on. It's all about growth and proliferation. This is potentially a natural change, all of which comes from slight damage to complex 1 leads to some kind of a quasi-aging program, and it doesn't require much. So that's roughly what I'm showing here. We have input of electrons into complex 3, maintaining a membrane potential, driving reverse electron flux into this, driving something that amounts to a reverse Krebs cycle, which is effectively driving cancer more or less as Warburg had been arguing a century ago, but in some sense, nothing to do with glycolysis at all, nothing to do with damaged mitochondria. It is a reversion to an earlier state, but that earlier state is in fact mitochondria as biosynthetic engines, not only as bioenergetic engines. So, if all of that is true, then you might think that if you were able to improve complex 1 function so that all this didn't kind of grind to a halt and start going into reverse, then better complex 1 function would be a good thing and would potentially slow down aging and have other beneficial effects. Well, this is something we've been looking at in flies, in Drosophila. And I'm going to show you some work that we've been doing. And there are 3 different fly lines that we've been looking at. And they are, the important thing to say is they're isogenic in their nuclear background, which means to say they've been inbred, they've been, there's various crosses you can do with what are called balancer chromosomes. But as far as we can make it, the genetic background is genetically identical. You can call all these flies identical twins. But they have different mitochondrial DNA. And again, this is, I'm not really a fly person, but you're able to effectively introgress the mitochondrial DNA from different populations. So there's no mutations here. This is simply mitochondrial DNA from a different population of Drosophila melanogaster, so it's all the same species, incorporated against a genetic background, which is isogenic. So we have three fly lines here. We have wild type, which effectively is this nuclear background with the co-evolved mitochondrial DNA. In other words, that mitochondrial DNA comes from that same population, and as far as we can tell, it's what it was meant to be. COX has a single point mutation in cytochrome oxidase, and BAR and this is actually this is from a population somewhere in the States. I forget where now, but it's a SNP. It's not a mutation. It's found at quite high prevalence in the populations, 30 or 40% prevalence in those populations in the wild. So it's not a serious problem for them. May have some benefits. And BAR is mitochondrial DNA from Barcelona, where there are 9 SNPs difference in protein coding genes. There may also be differences in the control region, which is much harder to sequence and to figure out what's happening. But these are differences in protein coding genes. And so what we expect to see then would be that the flies that are co-adapted, that the mitochondrial DNA matches the nuclear background, should be the best, and the ones that have got these mismatched mitochondrial DNA should be screwed up somewhat and not work as well. And the most screwed up ones should be the Barcelona flies, because they have the more genetic differences in their mitochondrial DNA. What do we actually see? We're looking at activity here. This is an assay called the T50 climbing assay, but basically you drop them down a tube briefly, and that stuns them briefly, and then they have a response, which is to climb up the sides of the tube that they're in, and we just count how long it takes for 50% of them to get back to the top of the tube. I don't know why they like to do that, but they like to do that. And what you can see here is that the bar flies, both the males and the females, do it really fast. They're back up to the top of that vial in no time at all. It takes the control ones and the COX flies somewhat longer, and the COX are better than the wild types here, which is surprising. On a high protein diet, we see the same thing. The bar females are a bit worse. The bar males are just as fast as they were before, and these other guys are even slower than they were before. So it seems like the Barcelona flies are the ones that are the fastest and most able to move around. What really took us by surprise is that they're also the most fertile. So this is on a control diet. This is the bar, this is the COX, and this is the wild type. And the bar flies are the more fertile. On a high protein diet, again, the bar flies are the more fertile, and the COX and the wild type are pretty similar to each other. And then in relation to this talk, really especially, this is longevity. The females and the males on the control diet, the bar flies live longer. This is the wild type, and right underneath it is the COX. So there's very little difference between the COX and the wild type for the females. And then on a high protein diet, everybody has a shorter lifespan on a high protein diet. But again, the bar flies do better on the high protein diet than either the COX or the wild type. Same for the males. The bar flies do better, actually do as well on a high protein diet as the other guys do on a control diet with more molasses and so on in their diet. These, the wild type and the COX, really don't like a high protein diet. They die very early, and notice here, they're already dying around about day 30. I'm going to come back to the importance of that in a minute. So this is the last thing we expected to see. The flies with the most mismatched mitochondria are more active, more fertile, and live longer. And I would like some of that. We also found in the larvae, so again, we're looking at the control diet. This is N-acetylcysteine, and I'll say more about that in a moment. It's an antioxidant. Turns out it's also a pro-oxidant and can cause reductive stress and can be quite toxic. And this is the high protein diet that I've mentioned. So this is developmental time. So the bar flies, there's not much in it, but they're slightly faster. They develop slightly faster. And this is survival. survival. So these guys survive slightly better as well. It should say proportion rather than percent here, but they're doing slightly better. So again, at the larval stage, they develop faster and they survive better. And this is mitochondrial DNA copy number, and it's a little bit lower. I never know quite what to make about copy number, but as a rule of thumb, if it's high, they're stressed. And these guys, these are the Cox flies on NAC, on N-acetylcysteine, they were stressed all right. They took a long time to develop, and their survival rates are relatively low, and they're showing signs of stress. So they really don't like NAC very much at all. The barflies don't seem to mind it very much, and they generally don't seem so stressed. So what about complex 1? Well, this is it for the larvae, and this is it for the young adults at day 10. And this is for females. For males, it was a less of a clear-cut picture. Now, these differences are quite small. That's the first thing to notice. So this is on complex 1 substrates, and this is the effect of rotenone inhibition, which inhibits complex 1. So these are two ways of asking the same question. And we're seeing at day 10 that there's a significant, in these are the adults at day 10, the barflies are using complex 1 somewhat more. They're also doing it with rotenone, and they're using alternative substrates, glycerol phosphate in this case, which is a substrate for complex 3 and which effectively short circuits complex 1. They're using much less of that. So that's also saying these guys are quite complex 1 dependent, and they don't like substrates from other places. And that goes for both the larvae and the young adults. So these are quite subtle differences, but it's important to realize these are about the only differences that we could see in respirometry, and we were using arose O2K with these, and I'll come onto that in a little more detail later on. It's pretty sensitive, and it's surprising how few differences we really see in respiration, but it's notable that the ones that we do see are in complex 1 activity, and the ones that are using complex 1 the most survive longer, live longer, they're more fertile, they're more active, and so on. So it really does seem as if complex 1 is a very good thing to have it functional. And this is almost the opposite. These are the same barflies, and when we gave them a very high dose of NAC, they were quite indifferent to a low dose, a 1 mg per mil dose or a 5 mg per mil dose, but this is a 10 mg per mil dose, and this was toxic. And what we found, rather surprisingly, this was what really started a lot of this work, 1 mg per mil was broadly beneficial. This is the proportion of survivors, so more of them survived with 1 mg per mil than with no treatment at all. These are the females, these are the males over here. And this was survival at day 12. And it really went off a cliff. For the bar females were almost all dead. 85% of them had died, 80% had died by day 12 on 10 mg per mil of NAC. These guys actually went off the same cliff a few days later, but there's a big statistical difference between them at day 12. And the males really didn't seem put out by NAC at all. Later on, they had a problem as well. They were shorter lived, but not dramatically so as in this case. So what's going on? This is the respirometry again. Female thorax and male thorax, oxygen flux. What we're seeing here is this is on complex 1 substrates, if I remember rightly, and the barflies have complex 1 suppressed by about 50% or so by 10 mg per mil NAC. These guys also had some level of suppression of complex 1, not as dramatic, but somewhat. But it stands out here. This is complex 1 contribution. So this is with rotenone. No, sorry, this is complex 1 over maximum, but this is the rotenone contribution. So different way of looking at the same question. And this is the control ratio, which is state 3 over, so just pyruvate and malate as substrates over maximum respiration. And all three of these indicate that the barflies on high levels of NAC are really not having complex 1 specifically suppressed. Maximal rates of respiration were quite similar. I'm not showing that here. And they were preferentially dying. This was the only difference that we could see that was apparently killing them. The males, which were not dying, did not have much reliance on complex 1 in the first place in comparison, and were not having it suppressed to anything like the same level. This is the oxidation state of the glutathione pool. So NAC is actually, it's a precursor to glutathione synthesis. I'm not showing it, but these flies had accumulated quite high levels of glutathione, but only in the case of the females on the female barflies on high doses of NAC was this beginning to oxidize. So the glutathione pool is kept in a normal reduced state for the other fly lines at that time point, and for the other doses of NAC, but these guys who died were beginning to lose redox control over their glutathione pool. Males, again, really didn't affect them very much. Now, this was the thing that I found most interesting. This is ROS flux, and you can measure it directly on an Ouroboros, which is to say you're using a fluorescent dye, which reacts with hydrogen peroxide, and so you're measuring the continuous production of hydrogen peroxide. Now, it's not necessarily there's all kinds of problems with using fluorescent dyes for measuring ROS, but the nice thing about the Ouroboros is you're able to look in real time. You can track oxygen consumption and ROS production at the same time in the same system, and therefore you can see if you add rotenone, how does ROS flux behave? Rotenone is going to inhibit complex 1, and ROS flux increases. If you have an uncoupler or whatever you do to the mitochondria, you can see the immediate response in ROS flux, and so you can see that this hydrogen peroxide changes in hydrogen peroxide flux are mitochondrial and behave in the way you would expect them to behave if you uncouple the respiratory chain or if you add rotenone. So what we're seeing here, in grey bars at the bottom, this is NAC, no NAC, 1 mg per mil NAC or 10 mg per mil NAC. These are the females at the top and the males at the bottom. If I remember rightly, I should have marked it. And this is just ROS flux on pyruvate and malate, if I remember rightly, and with nothing else. And it's remarkable how similar ROS flux is in all of those cases. Note, this is 10 mg per mil NAC. These are the flies that were dying. There is no change in their ROS flux, even though we know that their glutathione pool was becoming oxidized at this point. You add rotenone and you see an increase in ROS flux, and the increase in ROS flux again is pretty similar right across that board. You add antimycin A, which is inhibiting complex 3, and again, you see, as expected, another increase in ROS flux. We're beginning to see slight differences. There's less capacity of the respiratory chain to produce total ROS, but it's not a significant difference. So what is really striking to me here is that there's no change in ROS flux, really. It seems as if this suppression of complex 1, most ROS are coming from complex 1. It seems as if the suppression of complex 1 effectively means that there are fewer redox centers able to make ROS, and therefore, although each one might make more ROS, there are fewer of them, and therefore the total amount of ROS is maintained within some homeostatic limit. This is just a hypothesis. We haven't demonstrated that that's the case, but it's certainly consistent with our data. What that suggests is that suppression of complex 1 is a deliberate thing to do to maintain ROS flux within a homeostatic limit. Same thing goes for the males down here, slightly more variation with the males. This is all for the thorax, I should say. But it's very striking that ROS flux barely changes, even though complex 1 has been suppressed. So what might happen with age? What happens if complex 1 is being suppressed as we get older? Well, this is ROS flux at day 10 and day 31 in these fly lines, wild type Cox and Barr again. This is a high protein diet. This is a control diet. And this is the females on the left and the males on the right. I should have marked that as well. Slightly more variation here, but not much. I mean, this is a larger graph. And again, what really strikes me is how similar these values are. The gray, again, is just state 3 levels. There's no obvious difference. This is with rotenone, with antimycin A, and so on. So there are differences between them. Don't believe that any of them achieved statistical significance, though I need to double check. We're still processing these results. We've not published this yet, but we are writing up the paper now. So this is, I'm showing you really work in progress at the moment. But this would seem to suggest the same thing, that we are seeing a suppression of complex 1 with aging, which is maintaining ROS flux within some kind of homeostatic limit. That would be my working hypothesis. So I was quite surprised by the oxygen data. So this is maximum respiration, and this is day 10 and day 31, and you can see there's very little difference between any of these. Again, we can improve these graphs and don't think there's any significant differences here, but maximum respiration is pretty similar on a high protein or on a controlled diet in both of these, in males and females. This is maximum respiration, day 10, day 31. This is complex 1 linked respiration. This is essentially what I've shown you already. These bar females have got more complex 1 dependence, and also with high protein, they have a higher complex 1 dependence. I expected this to decrease, which it does, but what I certainly did not expect to see would be that Cox flies would increase their complex 1 activity with age. That was quite confounding. Also, there's really, they're slightly better. The barflies are better as young adults than say the wild type, but not obviously better than wild type at day 31. Remember, on the high protein diet, I pointed out to you that by day 31, a lot of the Cox and the barflies are already dying. They've just gone off a cliff. These are flies that are quite unhealthy, and yet they appear to have very good complex 1 activity. So we see this beautiful, good complex 1 function early in life is associated with really good outcomes, with high fertility, long life, lots of activity. But we look at complex 1 activity later in life, and we're seeing greater activity in what must be knackered mitochondria, but still ROS flux is maintained within some kind of homeostatic limit. That cannot be explained in the terms that I have been talking about. There has to be something else going on. And that something, well, what is it? So we've done lots of other work. This is metabolomics. Before I come on to what I think it must be, I'm just going to run through this very quickly. If this looks utterly confusing to you, good, it does to me as well. If it doesn't look confusing to you, please tell me what it means. I'm just going to, so this is differences, this is log 10 differences in metabolite concentrations. They've gone up where it's red, and they've gone down where it's blue, and where it's relatively, where it's fairly faded, then the change is fairly small. So the more intense the color, the greater the log fold change. Just to orientate you, because everything changes around, is based on trees of closest similarity. So this is bar flies on high protein diet at day 31 in each of the black boxes across here. So what we're seeing in the ovaries at day 31, this is comparing the high protein diet with the controls on a high protein diet, with the wild types on a high protein. Very little difference in ovary gene in metabolite concentrations. For testes, the metabolite concentrations have gone down pretty dramatically in barflies on a high protein diet in middle-aged barflies, day 31. For the male thorax, we're seeing generally an upregulation. For the female thorax, more of a downregulation. I mean, it doesn't make any kind of sense to me. That's showing it one way. Those are overall changes. All you can really see is that small changes in respiration, which is what I've shown you already, lead to big changes in metabolism, in the concentration of metabolites. This is not targeted metabolomics, so we don't really know what these changes in concentration actually mean in terms of flux. But we can put them into central metabolism and look at what these changes are. Here, again, I'm not going to go over this in any kind of detail. We don't know if these are really changes in glycolysis or gluconeogenesis. And I cannot tell from the context of it either. Here's the Krebs cycle. This is from glutamate, glutamine, and so on, various amino acids, and purine metabolism over here. And what we're looking at is barflies in the testes and the male thorax. This is day 10, day 31, day 10 with high protein, day 31 with high protein, compared to the wild type. And this is Cox. Day 10, day 31 on a controlled diet, day 10, day 31 on high proteins. I'm not going to linger on this because there are patterns, there are differences. These are log 10 differences, remember, so these are pretty big differences, but they seem to be doing more or less the same thing. And they seem to reflect that the flies which have got a mitonuclear difference to the wild type are behaving in a similar kind of way in the males, and this is the females. And again, there's nothing that I can see in this which is telling me what's gone on. All it tells me is that my understanding of metabolism is not nearly as good as I would like it to be. And maybe AI will be better at figuring out what these patterns mean. But at the moment, there's just so much flexibility in the passage of flux through the center of metabolism that it's very difficult to work out why are they so badly compromised that a lot of these flies are already dying. And this is gene expression. And again, we can see differences, but we don't know what those differences mean. These are pretty big differences. This is log-fold difference again. This is down, lowering expression, gene expression. This is increasing gene expression, and this is going up here is the p-value. So how significant that change is. So these ones, for example, are highly significant changes of down regulation. And these are kind of non-significant but big changes up regulation. We're looking at bar versus wild type day 10, day 31, high protein day 10, day 31. We can see at older flies, especially on a high protein diet, there's a change, but the older flies, we're seeing quite big changes in gene expression. The young flies, not very much. And this is comparing bar against Cox, again, with the young flies, very little to see, including on a high protein diet. With the older flies, day 31, sorry, this is day 30, I'm muddling up myself here. Oh, something funny's happened, sorry, I put the wrong thing in, this should be day 31. Anyway, the point is there are differences, but there's nothing in here that really explains why a small difference in complex 1 would lead to such a big difference in outcomes down the line. So I'm reduced to thinking about it. And what could potentially, in theory, explain these differences? Well, there's been quite a lot of rather beautiful work done in the last few years using cryo-ET. This is not my work. This is work from other groups. And this is all in Drosophila, and very often what you see is what people call onion mitochondria, which look like the membranes, the Christie membranes, turning into kind of onion skins almost, and we're seeing the same thing here as well. We're not seeing the normal Christie structure. We do see normal Christie structure in some of it, but we also see quite serious disruption in Christie structure. Now, the reason I think this is important is that the way we've traditionally drawn mitochondria over decades is something like this picture on the left here. And what we're seeing on the left is this is the intermembrane space. This is the matrix. These are the Christie. We're pumping protons from the matrix into the Christie space, and that Christie space is supposed to be continuous with the intermembrane space. That always worried me, because when we're visualizing membrane potential, for example, we're looking usually at dyes, and we're looking at lipophilic cations, and those lipophilic cations will dissolve in the membrane, and then they'll cross to this negatively charged space here and accumulate in the matrix, which is relatively negative compared to this. But if this is a highly positively charged, why on earth would they go anywhere near the matrix at all? It's possible they would, and they do, but it always struck me as a little odd that they would do that. What we've realized over the last few years is that really the Christie are typically closed structures, not always, and it varies with age. But when they're closed, there's an OPA1 complex down here, and the pumping is going from the matrix into this closed Christie space, which is much tighter. And so the intermembrane space, well, there is a little bit of a charge in the intermembrane space, but really very little there. So it's much easier for a lipophilic cation to accumulate in the mitochondrial matrix. And we also know that the ATP synthase is typically as a dimer, again, not always, but typically as a dimer, and it bends the membrane, as I'm sure you all heard from Wayne last week. These dimers will bend the membrane, and are partly responsible for the Christie curvature forming it all. And so we've got a very, very tightly ordered system. And with age, as I just showed you, if we just go back to there, we're beginning to lose this tight ordering of Christie structure. And there's a building literature on mutations in OPA1, for example, are associated with various age-related conditions. But if there's not enough of this Christie structure opens out and you end up with something more like this. Now, the reason that's important is that I already touched on it, that complex 1 is pumping 10 protons. It's unable to generate as strong a membrane potential as if you're using succinate or glycerol phosphate. In other words, there's just less power per proton to generate a high membrane potential. If you've got very closed Christie, it's going to be harder to pump into that space than into this much wider space where each proton is at the very least, there's going to be less of a pH gradient. I think the classic equations of electrochemistry need a look at here again, because they would normally say that it doesn't matter how large this volume is that you're pumping protons into. It's the same difference. If a charge crosses a membrane, it doesn't matter if you have an infinite space on the other side of the membrane. In my own mind, it surely must make a difference if you're pumping protons into a very constrained space compared to if you're not. Now, I'm sure many of you can correct me on this and know more about this than I do, but it strikes me that it's difficult to pump into a constrained space. There's hardly any water in this space. It's all structured water. It's very difficult to measure pH because it's all structured water. The protons almost certainly scuttle along, and they'll scuttle along down to the ATP synthase down here. So there's a structure to what's happening. Before I come back to that structure, I want to introduce some other work that we've been doing recently, which is on anesthetics, because they give some sense of how complex 1 functions in relation to these charges and what might actually be happening. So one thing that's been known actually for some decades is that volatile general anesthetics will suppress complex 1. And this is again work with an Ouroboros, and this is showing isoflurane and sevoflurane, and this is with complex 1 substrates. And indeed, we get about a 50% suppression. We're giving quite high doses. We're giving multiple doses of anesthetics, these titration steps each time we're giving, well, varying amounts, but usually 400 micromolar of anesthetics into the Ouroboros. So it's a fairly large dose that they're getting, which would probably kill real flies. It's interesting, though, that the membrane potential, this is relative membrane potential, this is the reciprocal of the fluorescence of the dye, and we're not converting it into membrane potential units because it's very difficult to know what they really mean, but we're certainly seeing an increase in membrane potential. That's complex 1 linked respiration. This is maximal respiration, where the suppression of maximum respiration is much less and is entirely attributable to complex 1 suppression. And we're seeing again an increase in membrane potential, which is perhaps harder to explain why it would be larger than that. I'm unable to do that at the moment. Down here, we're seeing the same thing, except we realize that ethanol has all kinds of effects on mitochondria and on pretty much all of these processes. And I'm not showing you any of that, but this is excluding ethanol. This is with the neat anesthetics, and we're seeing the same effects, which is a suppression of complex 1 with both isoflurane and a lesser suppression of complex 1 with sevoflurane, but an increase in membrane potential with both of them. And again, this is a fairly high doses, but it's neat this time. So we're seeing an increase in electrostatic membrane potential despite a decrease in electron flux. This is the same thing again, and again, we're seeing here a greater suppression of complex 1 with isoflurane and sevoflurane. But the only real way to explain why you would see an increase in membrane potential is if the ATP synthase goes into reverse and starts pumping protons to maintain the membrane potential. So we can inhibit the ATP synthase with oligomycin. That does what you would expect it to do. It collapses you can no longer have any electron transfer, so respiration collapses. But when we look at the membrane potential, this is what we're seeing, a gradual increase in membrane potential when we add oligomycin. we block any flux of protons going in any direction through the ATP synthase, and we see an increase in membrane potential back up to the leak state from the beginning. That means that we must have some electron flow, and it must be capable of generating some membrane potential because that's the only way that it would possibly go up again. Luckily, with the Ouroboros, you're also able to look at ATP synthesis, and this is a relatively new way of doing it. It's with magnesium green. I'm amazed that this works at all. What you're really looking at here is fluorescence of magnesium green, which depends on magnesium and the concentration of magnesium. The dissociation constant between ADP and ATP is slightly different, but these results are quite dramatic anyway. So this is effectively where it's pointing up, we have more ATP than ADP, and where it's going down, we have more ADP than ATP. So we're seeing a collapse in ATP synthesis with the first dose of anesthetic, and the second dose slightly bigger than the third dose. I'm not sure what's happening there. Same thing with sevoflurane. We're seeing an immediate collapse. As soon as you give an anesthetic, ATP synthesis collapses. And that is completely consistent with the idea that the ATP synthase goes into reverse and starts pumping protons at the expense of ATP. So all of that's a rather beautiful story. And this is ROS flux, again measured at the same time. We see a rather paradoxical initial decrease in ROS flux followed by an increase. We see the same thing with sevoflurane. Well, it turns out that the dye that we're using to measure ROS flux is affected by the anesthetic as well. So there's two runs here. I'm not going to go through the details of those, but one of them is in tissue and one of them is just the dye plus the anesthetic. And we see a decrease in fluorescence just with the anesthetic alone. So that's what we're seeing here, which means that this increase in ROS flux is probably greater than we're marking, but we don't know exactly how much. So this is again considered to be a problem with giving anesthetics to people over the age of 70 or 80 or something, is that they can be quite damaging. One of the reasons for the damage has been postulated to be ROS flux. And what we're showing here is that anesthetics suppress complex 1, that reverses the ATP synthase, which now increases membrane potential, and that increased membrane potential generates increased ROS flux. So all of that makes some kind of sense and would suggest that the way that anesthetics work is by just collapsing ATP availability. But there's some mysteries, and they're quite interesting mysteries, and I think they say something about aging and things as well as consciousness and anesthetics. Nobody knows how anesthetics work in the sense that they have completely different structures. And the only thing that they have in common seems to be that they will accumulate in lipid environments. The greater the lipid solubility, the more potent the anesthetic is. But there's all kinds of madly different structures. And the idea that they would simply bind to some pocket in some particular enzyme, or the binding pocket in complex 1 or anything like that, is just not consistent with this great variation in structure of anesthetics. And there's some beautiful work from the last couple of years on chirally induced spin selectivity. Now, this was work that's been done by Luca Turin and Ron Neyman. And effectively, if you transfer electrons through a chiral medium, the earlier work on this was done literally with chiral molecules that were absorbed onto the surface of an electrode. And you pass electrons through that chiral surface, then you polarize their spin. And anesthetics have been shown, and multiple anesthetics have been shown to disrupt that spin polarization, so you lose the spin polarization. This happens in respiration. It's been shown to happen in respiration, that the transfer of electrons to oxygen is slowed down, apparently by this loss of spin polarization. Now, the interesting thing is, why and where would electrons be spin polarized in respiration? So Luca Turin, who I've talked with about this kind of work, and we have a grant together on this, which is still under consideration, but he's thinking in terms of electron transfer all the way through to oxygen, because oxygen is a diradical, and so prefers to accept electrons in the same spin state to reduce it. And so Luca is seeing this in terms of the entire respiratory chain and transfer of electrons to oxygen. From my point of view, though, there's a bunch of questions about complex 1. Why does it have so many iron sulfur clusters? Why is it quantum tunneling over such a long distance through these iron sulfur clusters? Is it to increase spin polarization of electrons within complex 1? But if you were going to do that, well, why would you do it? Because when they get to the quinones, you then lose that spin polarization and it's reduced to a quinol. There's nothing paramagnetic about quinols. So you then have to regenerate it all at complex 3, in which case, why bother setting it up in the first place at complex 1? So there's a bunch of interesting questions there. And there's a long literature on people with mutations, for example, in complex 1, and various animal models. Mutations in complex 1 lead to greater sensitivity to anesthetics. And that's not true for complex 3 or complex 4 or complex 2 or the ATP synthase. There's something specific about complex 1. If it was only a collapse in ATP synthesis that was leading to unconsciousness, you would have thought that mutations in complex 3, especially if that was regenerating spin polarization, would make you just as sensitive to anesthetics as those in complex 1, but that's never seen, that's not known. So there's something special about complex 1 in relation to spin polarization and the mechanism of anesthetics. It's not entirely clear what that is, but I suspect it links to electromagnetic fields, which is why I'm telling you about it. So I've already shown you this structure, the Christie structure. We have most of the complexes are embedded in the sidewalls of the Christie, including complex 1, and we necessarily have a proton circuit, which is likely moving charge is going to generate some kind of field. And there's some interesting, beautiful earlier work. This is from Doug Wallace, his group anyway. Martin Pickard, I think, was the senior author on this. And what's beautiful about this is the way the Christie line up. This is 2 separate mitochondria with the membrane dividing them, but the Christie membranes are lining up between those different mitochondria. And these are various knockouts of mitofusins, which are responsible for the docking of mitochondria. So you can prevent the docking of mitochondria and you still see the Christie lining up in the same way that they do before. It's really a dramatic effect. And it looks a little bit similar to the magnetic strip domain structure that you see, for example, in manganese aluminium thin films. This is with magnetic force microscopy. So it seems to me as if there's some tantalizing evidence that there are electromagnetic and probably magnetic fields going on in mitochondria. And if you think about the way that complex 1 is working, if we have all of these Christie lined up on this side of this Christie, we have a current of electrons going towards the surface. Remember, if I just come back a couple of slides, it's almost an L-shaped molecule. This electrons are being transferred towards the surface of the membrane, down through here to the surface of the membrane, almost perpendicular to the membrane. So we would have a current of electrons going perpendicular to the membrane in multiple complex 1s on the same flat surface, generating a magnetic field perpendicular to that, which should rope in all the complex 1s on that surface. Same thing should apply to all of the surfaces of complex 1 across here. And we have a proton current going down, which should generate a perpendicular magnetic field going across, which should link all of these Christie together. So we would have synchronized electron flow through complex 1 in all of these Christie facing the same direction. Soon as the electrons pass on to quinones, that field will cut out completely, and all the complex 1s on the other side of the Christie are now free to push their electrons into complex 1 in the opposite orientation. So we should have an oscillating magnetic field generated by parallel Christie in mitochondria as a result of electron transfer in complex 1, and linked together by a magnetic field at right angles to that because of the direction of proton flux, which is not changing and therefore would always be the same field in opposite fields in opposite directions. So it's perfectly possible in principle for mitochondria to generate magnetic fields that could at least explain that orientation of Christie between mitochondria, but it could operate over much longer distances. And my own back of the envelope calculations suggest that we could have magnetic fields in synapses, for example, over distances of up to about 10 micrometers, so 1 to 10 micrometers in the range of hundreds of microtesla up to around about 1 millitesla. So above the background Earth's magnetic field, sufficient to do work of some sort. I'm touching here on Mike Levin's work. Mike has, as you all know, I'm sure, has done lots of work, not really on fields as such. He's talking about electrostatic voltage maps across the plasma membrane of hundreds of cells, which are linked by I forget the name for them now. The channels between cells. So they're all sharing the same voltage across quite wide areas. And this, I think he's persuaded you all, and certainly me as well, that this kind of voltage mapping has a lot to do with development, and which goes on to be the head, which goes on to be the mouth, and so on, not only in planarians, but also in more complex things like frogs and possibly in us. And more relevant to this talk, it's also possible to distinguish tumors on the basis of their plasma membrane voltage. And this is very much to do with the ion channels in the plasma membrane. And so in this case, we're seeing that the cancer cells are depolarized and the rest of the tissue surrounding it is hyperpolarized. So this is at the level of the plasma membrane. Everything I've talked about so far has been to do with mitochondria and the flux through mitochondria and the centrality of mitochondria to cancer and to metabolic flux. And the question is, and this is something I've started to address with Mike, is how do the mitochondria talk to the plasma membrane, and how instantaneous is that? And most people who've worked on these kind of questions will either think in terms of metabolic biochemistry directly, or possibly the cytoskeleton. And signaling through the cytoskeleton. My question really is can we measure magnetic fields that stretch between the mitochondria and the cell membrane? In principle, they will interfere with the opening of voltage-gated channels in the plasma membrane. My calculations as to how strong these fields could be is sufficiently strong to interfere with voltage-gated channel opening, and therefore with the polarity of the plasma membrane. And the point in relation to this talk is that if we're seeing these structures breaking down with aging, and we're losing the parallel Christie structure, and we're losing the synchronization of complex 1, we're losing the magnetic signaling as we get older. How serious is that? How much of an effect is that? I really don't know. I mean, the fact that we remain pretty much conscious and alert and our systems function until the end says that, until quite late, says that this is not necessarily the only thing that's going on. It's probably there are more conventional ways of doing it, but it would be very surprising to me indeed if there were not direct and immediate signaling mechanisms between the mitochondria that are the interface really between the outside world and metabolism. This is where substrates and electron acceptors are interacting with the outside world, and the plasma membrane, which is the physical interface with the outside world. So we have, to put it in kind of Carl Friston terminology, we have two Markov blankets, one which is the plasma membrane, one which is the mitochondrial membrane and inner membrane. And those Markov blankets need to talk to each other in some way. And I am increasingly thinking that that way is through fields. I think I'm going to just stop there. This is kind of a summary slide, but you can read it as well as I can. But I think it's time to move on to have a general discussion, and you can take apart some of my physics here, please. Thank you.